Welcome to our Applied Math Colloquium. We have Dr., and I don't mean just PhD doctor, I mean real doctor, Jake Scott, here from the Cleveland Clinic to talk to us today. I only met Jake yesterday, but we have known each other a little bit on Twitter for several months now, and through Skype. We've been working together along with Bertrand and Tina Loeffler here. And so yeah, we met on Twitter because he goes by the handle Cancer Connector, which sounded interesting to me. And as I started reading his blog, I ran into a post about the application of evolutionary dynamics to cancer, specifically evolutionary graph theory, which seemed interesting. And then that led to some emails, and pretty soon we were working together. But in the course of that collaboration, I learned that Jake is, you know that beer commercial about the most interesting man in the world? It's actually him. So he has had, in his earlier lives, time as a high school teacher, where he was also a wrestling coach. He was in the Navy and worked in connection with nuclear submarines and was on duty at the time of 9/11, when it was not really a joke to be, sitting at, you're not actually in control of the nuclear weapons.
No.
No.
And well, we were later that day.
Okay, but anyway, so there's that, and then medical school, and later specializing in radiation oncology, where he now treats patients 1 day a week. But also he made time for a DPhil in mathematical biology at Oxford, and he's going to tell us about his work with his team on a very important problem of how people can become resistant to chemotherapy drugs. So it's about the evolution of resistance. Thanks.
Hey, yeah, thanks all for coming. This should be fun. I'm going to give a sort of wide-ish ranging talk, and I think I'm going to have 1 foot in the biology and 1 foot in the math almost the whole time. And so, by nature, I won't be able to go, and also maybe because of my own skill set, I'm not able to go as deep on either side as maybe some people are, but I think what I do offer is the ability to straddle it meaningfully and wholeheartedly. And so I think that the most fun part about this job is meeting folks that are smarter than me, which is everywhere I look. I think we all have something to teach each other, and I think that that's the most fun part of this job. And so I think that what I'm going to show you is my attempts to understand the biology through the lens of mathematics, and my attempts to advance the mathematics through the biology, and back and forth. And it is a team effort. Everything I show you is going to be done by me and friends. And please share whatever you like. The whole point of doing science for me is to share the science. And in particular, I think that human disease is something that we ought to all collaborate on. So if you see something that you like, tell me. If you see something you don't like, tell me. If you see something you want to work on together, tell me. If you see something that I should stop working because it's solved, tell me. And at any point during the talk, please, just yell at me or raise your hand or whatever, and I'll stop. It's going to proceed in sort of 2.5 parts. So the first 40 minutes will be 2 very highly connected parts. I'll stop in the middle. There'll be a math part and a biology part, and then I'll stop before I can move on with an evolutionary game theory part at the end, to the last sort of third. So I'll try to make breaks in the middle where there are natural times to ask questions, but please, otherwise, just shout at me. So that's what we're going to talk about. So first, I think that probably most of us in an applied math room believe sort of about how we should go about modeling stuff. And I think the most important parts here are down here, and that more complex is not necessarily better. I think it's a misconception. My biological colleagues are always like, well, why didn't you include this other thing? Why didn't you include this third thing? And I think really stripping things down to its bare essentials is most important. And I think focusing on both what we know and what we don't know, and also talking to our friends. The dialogue in science is for me the most fun thing. And so there's sort of my 2 most important tenets are, all models are wrong. Everything I show you today is wrong, for sure. Please point out why. I probably am wrong in more ways than I even know. But if they're useful, then that's all that matters. Even if it's wrong, if it helps advance somebody's thinking, then I've achieved my goal. And then this simple but no simpler idea. So what else? So here's what we're going to talk about. These are real pictures. This is not my patient, but I've seen patients like this. This is just a very striking figure. So on the left-hand panel, you see a front and back view of a mid-30s gentleman with a special kind of melanoma. It's all over, as you can see. Most of these cancers start in one place, a funny mole that goes crazy, and in this man's case, it's spread all over his body. He had a specific mutation within his melanoma called BRAF600E, which is just an index for a specific base pair, and there's a drug that works really well against that. And this is that gentleman after 3 months. Looks like a normal dude. 4 months later, it returned and he died. We see this not occasionally, this is every time. This is how it happens. This is what happens. This happens in lung cancer, sorry. This happens in lung cancer, melanoma. I'm going to talk about lung cancer today. This happens in antibiotic resistance, not the spots like this, but the same idea. Drug works, drug doesn't work, drug never works. This happens in tuberculosis, which is a third kingdom. This happens across the kingdoms of life. When it comes to an evolutionary pressure, evolution figures out the answer and carries on, just like we do. We're faced with adversity, we figure out the answer, and we carry on. I would submit to you that this is the fundamental problem in medicine today, both in cancer and infectious disease, which are basically the #1 and 2 causes of death. So that's why I'm focusing on it. I think it's important.
Quick question.
Please.
So, would he have lived longer had you never given the drug?
No, he would have died shortly after this, within weeks probably. So, but what did we give? We gave a 6-month increase, maybe 9 months, which, you know, I'd take 9 months more with my kids if you offered it to me. And this gentleman had 9 months more to get his affairs in order and take care of stuff and live. But no, this did not speed the his demise. On the contrary. Okay, so a lot of times people think, I'll just add more drugs in. If there's more than one mutation, or if there's this idea of heterogeneity, I'll just add another drug. The problem is that combinations of cancer drugs can be really toxic. You give people too many drugs, you kill them and their tumor, right? So you succeeded in killing the tumor, but you also can kill the patient. So this is sort of not always a good strategy. It does work on occasion, but also the potential pool of drugs, and we're going to talk about bacteria to start and then cancer to move on, but I think I'm going to try to move back and forth between the scenarios as analogs. There really are. And once I present the mathematical model, it's abstract enough that it could be either. And so what we're going to think about in this first part is, what if instead of spending all our time looking for new mutations and designing new drugs, what if we instead thought differently about how the drugs work and recharacterize the ones we have in a sensible way and think about using them in sequence as a way to extend their efficacy? And I'll explain what I mean in the coming slides. So the next slide is a paper that, this is an ad for what I do. I call my lab theory division, which is a joke about joy division. And as a physicist, I sort of attack problems with whatever tool I think is necessary. And so we use a whole range of stuff, from cellular automata to what I was classically trained in, and ODEs and Markov chains. And I'm not afraid of dipping into the biology a little bit either. But I think that the first model we're going to talk about is going to be a Markov chain, which I think should be highlighted as like a blue circle or something. Oh yeah, this video is playing. Sorry, I shouldn't have animated it. So, okay, here's what we're going to do. I'm going to explain this slide in a lot of detail, because you're going to see matrices that look like this a lot in the coming 15 or so slides. This is a paper that I didn't write. This is from 2013 in a nice-ish journal. And what the folks did was this. They took E. coli off the shelf, which you can buy from a company. Or you can just go and get it in your hamburgers, whatever you like. And what they did was they took this bug and they made 15 plates of the same bug. So you hope they're all the same, but maybe they're not exactly the same, but it's as similar as you can get. Same stock, 15 plates. And then from each of these drugs, all of which have names that I spent a decade memorizing, so I'm going to tell you a few of them to impress you. Nitrofurantoin, triamcinolone, rifampicin, takes you aspirin, call me in the morning. They then evolved resistance to all 15 of these. So now imagine 15 plates of the same bug, and then we apply 15 different selective pressures to each plate, and then evolve them forward in time. So we start with all the same creature, now we evolve 15 new creatures that started from the same thing. Then we ask the following question. So now we have these 15 new creatures, And now we look at all the other N-1 drugs that they weren't evolved against, that their friends were, and we test that new creature against all the other drugs. So for example, this square represents a bug that had evolved under amikacin and its sensitivity to ampicillin after having been evolved to this, normalized by the wild type's sensitivity. So normalized by the one I bought off the shelf. So what I'm asking here is, how does the evolution to the first one change the sensitivity to the second one compared to how it would have been? So now you can look at this and say, okay, so red is really bad. So let's take a red one, for example, here. So after ceftriaxone, you should really not give cefprozil, because it's 32 times more resistant than it would have been if you had never given the ceftriaxone. Make sense? And then the opposite holds true. So after chloramphenicol, you should definitely try amikacin, because it's way more sensitive than it would have been. Good?
But shouldn't the whole diagonal be red?
Yeah, it should be. But biology is messy.
Wait, what does it mean?
It means that the whole diagonal should be approximately red, and they are approximately red. So they are more resistant, but they're not always all the way resistant.
I see. So we never, okay.
Because like, if you train on AMP, somehow they're more resistant to impacts than the AMP.
Sorry, which one?
Yeah, like
4 and 5, or 5 and 6.
Yeah.
Because the diagonal is less resistant.
Oh, it's like, yeah, exactly. So there's something going on. Please, someone else had their hand up? Yeah.
So resistance here is strictly something negative, because before you were also talking about poisoning the patient together with the...
So here, this resistance means a very specific thing, and what it means is that they measure something called minimum inhibitory concentration, which is a thing that's very standard in microbiology, and it is exactly this following measurement. I have the bugs, I drip on, so what you do is you get 15 plates, and you drip on nothing, control. You drip on a half, you drip on 1 concentration unit, half, 1, 2, 4, 8, and you go all the way up, and then you find which one is the minimum concentration that stops growth. That's the assay. And then the question is, so then when it says 32 times, that means it's that many fold times change compared to the baseline. Okay? Again, not my work, but this changed the way I think about things a lot. It's pretty cool. This over here is just a count of how many are more or less resistant. So after amikacin, you have a certain amount of more sensitive or resistant. They then did the following thing. They said, okay, cool. This is the extrapolation they made next. And I think if you get a hairball diagram like this, you like go up by 5 impact factor points in your journal. So I think that's half the reason they did this. What this says is that they said, okay, well, what if I know that after chloramphenicol, amikacin is really good? What then? So what happens then if I fail the amikacin that I secondarily gave my patient? Oh, easy. You just find amikacin and you look for a blue one. I'll get this one. Then what happens when I fail this one? Oh, easy. You just find that one on here, and then you find another blue one. And so this network here gives both cycles and complementary pairs, and this is a digraph now, which gives you the idea that you could sort of go through this network and constantly have more and more sensitivity. Please.
There isn't really any memory beyond the last step.
Yes, I think there probably is. But in this work, there's not yet. But I'm going to address that directly. Does everyone get that? Yeah. Okay, so that paper, you guys have ever had a moment in your life where you read a paper and nothing is the same afterwards? Have you ever had that moment? It's like the best part of science, right? So I read this paper sitting, I had a baguette with goat cheese and tomato and fennel, or rocket, sorry, they call it that. And I was sitting at the base of this monument outside the Mass Institute in Oxford when I read this science paper. This guy from Brown, Dan Weinreich, wrote this paper that would change my world. And it's going to, the reason I'm showing you this is because it tells you why I built the mathematical model that I'm going to present in the next slides. He took, again, E. coli. He took the 5 most important genes, the 5 most important base pairs within the most important gene that codes for resistance to a family of antibiotics. Okay? It's a mouthful. One gene that's the most important when it comes to fighting off the most important selective pressure, 5 spots within that gene, all of which individually increase the effectiveness of that gene. So anytime E. coli mutates one of these spots, it gets more fit in the presence of these drugs. So what he did was he and his team took the E. coli and they used the magic of molecular biology to flip each of those mutations that's possible to get 2 to the 5th, 32 new strains, each of which had one of the combinations of those 5 bit flips. So now he's got 32 strains. He does that same experiment I described to you earlier. He measures the minimum inhibitory concentration and just wrote it down. So now, if you wanted to draw a network that described all 2 to the 5th things and how you go from one to the other, it's just a hypercube, because you now have a bit string and you're flipping single bits, you get a 5-dimensional hypercube. And he asked the question, if evolution only allows you to go from less fit to more fit, how many ways can they percolate through this hypercube? And the answer is not 120, which is how many it should be if you're going from 0, 0, 0, 0, which is the least fit, to 1, 1, 1, 1, 5 1s, which is the most fit. And before this paper, people thought every single advantageous mutation should go uphill. It turns out that's not the case. It turns out there's an immense amount of ruggedness in even this tiny simplified version of an evolutionary landscape. And this is really the first experimental evidence of that, and it was only recently. And so we found there's only 18 ways you can get through this small landscape. There was a second paper from a group at MIT by a guy called Jeff Gore, who's actually a physicist by training, who now does really actually cool stuff that I'm kind of a fanboy. He did a very similar thing, but he took an extra drug. So he took the same 32 lines, he stole them from his friend Dan, and then he added a second drug, and he asked the question, in the presence of 1, if I evolve under 1, and then go to the next, what happens? And what he found is not only are some of the pathways through the hypercube not transmissible because of evolutionary valleys, but he secondarily found that there were some pathways that after a second drug you couldn't go back. There were some places that you could get stuck effectively. And so these two papers together led me to the hypothesis that can we irreversibly steer evolution with one drug such that when another's applied, the evolutionary trajectories to that spot of highest resistance that killed that patient 2 slides ago becomes inaccessible. So that's the central hypothesis. Does that make sense? So graphically, like this, obviously we all know that this space here isn't the way a hypercube looks, but I don't think I probably need this slide for this audience, but this makes more sense to some other audiences. The idea being, if we know something about the peaks and valleys of a landscape, can we use the knowledge of those peaks and valleys to shepherd a population through a landscape such that maybe you're trapped in one place, that it goes from being very, very, maybe we use this one and then give this one second. Maybe we can use a single drug to bring a population, maybe not even kill it, but just to shape it into a situation such that a second drug becomes incredibly useful. Maybe even a drug that doesn't seem to work. So, we wrote a paper, which you can read in detail, but I'm going to present now. So this is Dan. He looks suitably concerned. This paper also got us an error number of 3, which is awesome. Turns out my next door neighbor in Florida, who is now one of my patients, which is odd, his dad is a theoretical computer scientist at Ohio State. He has error. So we got him to be a co-author. Anyways, so I'm going to show you, I'm going to walk you through some of the results from this paper. The rest you can read on your own if you're interested. So what we do is we take this idea of a fitness landscape, and I think everyone here probably sees this more clearly than most, but if we have this idea of a landscape, which since the 30s has been represented nicely as a hypercube, we now have a genotype space that is simply a string, a binary string of whatever length we like, and now we're going to define a fitness function that maps from that string to a real numbered fitness. So what is this real numbered fitness? Well, in the bacteria stuff I showed you a little while ago, it could be that minimum inhibitory concentration, but maybe it's something else. Maybe it's the growth rate, maybe it's whatever you like. It's fitness. I think this is one of the hardest things in the study of evolution. I think most people agree that fitness is the thing, but nobody can agree on what fitness is, which is hard. So here we're just going to call it a real number. It's awesome. And then we're going to...
I don't want to be annoying about it. But,
no, please do.
But with the A, C, G, and C, what is the zero?
Yeah.
Well, just tell us how we should think about that.
So in the context of this model, let's think about the zeros and ones instead of being base pairs. Let's think about them being individual genes, and let's think about a one being it.
Oh, it's a whole gene.
It can be.
Okay.
Otherwise, I should. You're right. Go 0 to 3.
Right, so a zero is what, gene on or gene off or what?
Wild type.
A wild type or
And then one is mutated.
allele. Okay. Or not allele, it's gene.
Allele. You can write that down.
Yeah.
Again, an abstraction. And you're right. If I wanted to do this for base pairs, it would probably be
You're saying we're not
0.
doing it, so it's a mutant version of the gene.
Yeah, so we're gonna go back in time to the 30s when they didn't even know about the base pairs and they just knew about mutant alleles or regular alleles. Okay. And we're gonna invoke the strong selection mutation assumption, which is a super common assumption in the study of population biology, and in particular, it tells us that selection is strong and mutation is weak, which means that as mutations occur, selection makes it such that that mutation fixes instantly. So what we have is a monomorphic population. So this is a great assumption because it lets us do analysis, but it's actually a terrible assumption when it comes to the real biology. And I will relax it later, I promise. In this talk, not just like in my life.
What is monomorphic?
One type. So the population will always be everybody is this type, or everybody is this type, or everybody is this type. Later on in the talk, we'll relax that, but for now, we're going to have that. And now what we're going to do is we're going to model the population is all. That's what it says here. All the population has the same genotype, and it's periodically replaced by a fitter mutational neighbor. And what we're going to do is we're going to do the same thing mathematically that we saw experimentally a few slides ago. We're going to make a hypercube of size n, and we're going to ascribe or prescribe a fitness to each vertex. So this one has fitness 5, and this one has fitness 4, whatever. And so then we have this object, which is now a digraph, because we're going to say that arrows only point from low to high. So this is just like that picture we saw in the science paper. The next thing we're going to do is we're going to assign a probability transition, a transition matrix or a transition function, such that we can tell if we know the fitness of a given node and its neighbors, we can then figure out what the probability of going from one node to any other node is in a given time step. We're also going to say that when things get to a peak, they don't go anywhere. Maybe a bad assumption, but that's what we're going to stick with for the moment.
So, where did the fitness values come from?
Right now, they're arbitrary, but we're going to use data in a moment.
I'm just wondering, so in particular, they are not related, they're independent of the transition matrix. Because one would think if, like, I have directed arcs to 3 different points with really high fitness, then my fitness should also be fairly high because I'm going to evolve into something with high fitness.
I guess I disagree. I think, well, at least in the setting of this model, I wouldn't agree. I would say that, so first of all, I'm defining fitness simply in this case, it's simply a probability of something sort of coming toward you, really. And so the probabilities are mapped from the fitness, not vice versa.
Got it, okay.
Mm-hmm. Okay.
Could you read the formula or at least explain what's in the numerator and denominator?
Yeah, so this is the fitness of where you're going, fitness of where you are, and this is normalized by the fitness of everybody. And then there's a little r here, which we take in this case to be 1, which means it's biased by everybody, rather than, if you can imagine, if you put this to a 0 or an infinity, it would change the way this works. So one of them becomes steepest gradient ascent, and the other one becomes completely random. Any uphill is equally, so put a 0 there, and you can just have, you can go any direction. So if you have 2 uphill choices and 1 downhill, it's 50-50 for the uphill choices. In the R equals 1 case, if one is really, really uphill and one's a little uphill, you're more likely to go the really, really uphill. And if R is infinity, you just go uphill the most. So it actually, the results here are robust to changes in R, which I won't show you, but they're there. You can read about them if you like. So now we have the ability to understand discrete time population dynamics in this model. So if we have some initial population distribution, which we'll call mu0, and then we have a time step, which we'll call this, we just basically have mu at time 0 or mu of time t plus 1, we just have it as matrix multiplication. So we have some population vector, some transition matrix, and by repeated multiplication, we can iterate the evolutionary process going forward. So we have this discrete time, and then finally, we can figure out the steady state limit of this by taking the limit of p to infinity, or sorry, the exponent to infinity, and we get this steady state behavior. You can also prove that it doesn't have to go all the way to infinity. We also show that. But the point is that we now know both discrete time population dynamics and steady state dynamics. So we reduce this all to matrix multiplication, which is nice because it's fast. But it's also fun because now we have the entire sort of evolutionary effect of a given landscape, which is a drug, in a single, what we call limit matrix. So we've encoded the entire sort of evolutionary process in this one matrix. So that's steady state. So the first cute result here is that evolution doesn't commute, which is kind of interesting. So if you take 2 different drugs, for example, drug F and drug G, we know that unless these matrices satisfy very specific requirements, that we can't flip the order because matrix multiplication doesn't commute except for very specific reasons or times. And further, we actually also simulated this for millions and millions of NK landscapes. And basically only landscapes that are single peaked and are the same peak, which makes sense, right, if you all go to the same peak. Doesn't matter which one you do first. But the important thing here is that for me as a clinician, this is telling me that beyond what drug I give and beyond what dose of the drug I give, the order in which I give them matters. And this speaks directly to your question about the memory in that experimental model, right? So if you extrapolate to the 3rd drug, now if you give A and then B and then C, this result tells you that that's very different than giving B and then A and then C. Or could be. Because your initial condition is going to be different after each evolutionary ordering.
Remind me what the star means on P. Is that
That's the, so we take that landscape and
it's
it's
the
the limit.
ultimate steady state.
Yeah, it's the limit. Exactly. So we call it the limit matrix, which is the
So that limit matrix depends on R too.
It does, but we're going to have R equal 1 for this whole thing.
And I guess how does the steady state maybe vary with R?
That's a good question. We looked at the results I'm going to show you on the next slide. They're qualitatively robust to changes in R, but they are quantitatively different and effective. But we didn't go any further than looking at qualitative changes. Because it made the reviewer happy and he went away. You don't look pleased yet.
So these are not these market chains that absorb these states, right?
Yeah.
Okay. So you're talking about the probability of landing in a given absorbing state with those P stars.
Yeah, basically.
Okay. So that will depend on conditions.
Absolutely. Yeah, absolutely. That's why we said, yeah. So here, if you did know that these are exactly the same, then this actually is okay. But knowing that initial condition is actually really hard. So the results I'm going to show you in the next few slides, I'll assume a perfectly spread out population, which is sort of the null. And then usually when I give this talk, it's to doctors, and so this is something that every doctor in the world would recognize. This is a screenshot from a website we all cheat with called UpToDate, which if you don't know what you do in a patient's room, you ask to use the restroom, and then you look really quickly on your cell phone to the website called UpToDate, and then it tells you what to do next. But the thing is when a patient comes in with methicillin-resistant Staph aureus, which Stephen had, you can do any one of those things next and be right. And I think that this is a call to suggest that maybe there's more to think about. So order matters. So the next thing we sort of did was we looked at the following experiment. And I'm going to flip to the next slide, and then I'm going to come right back to this one. So we did an experiment in which we used 15 landscapes that somebody else measured. Okay, so now somebody else went through that same experimental setup that the guy did at the beginning, but instead of one drug, he used 15 used 15 drugs, and characterized 15 landscapes for E. coli with a 4 landscape. So we set up the following experiment. We asked, for each of those 15 drugs she measured, can we take and look at the initial distribution, knowing nothing about it, and apply drugs in all possible orders for up to 1, 2, and 3 drug combos, and then ask the question, are we better off or worse off than we would have been if we just applied the first drug? So this is sort of like the 3-step process of that experimental picture at the beginning. Make sense? The next 2 slides have a lot of numbers on them, and if you're far away, just close your eyes. I'm going to highlight the ones that are important, because basically what I'm going to show you is an exhaustive search of this space. And so, sorry. So if you exhaustively search, it's a terrible slide, but I can't figure out a better way to go through it. So I think I highlight something in the next version of it. No. So if you exhaustively search all of the combinations of 2, 3, and 4 drugs, what you find is that giving it an arbitrary order, we may be inadvertently selecting for high resistance. So overall steering, 61% of the time, we are doing worse arbitrarily than if we had just given the first drug. If we give 2 drugs first arbitrarily, 70% of the time we're worse off. And if we give 3 drugs arbitrarily, like docs do, we're worse off more than 70% of the time than if we had just given the first drug to begin with. And this sort of speaks to, this is what we do. You get an infection, and you go to your primary care doc, and he gives you whatever drug the most recent salesman was selling. It's like, oh, well, he gave me a sandwich, so I'll give you moxifloxacin, because they're all equivalent. But the scary thing is, and then what happens in the hospital is we all get these patients who've now had 3 drugs in a row, right? And so now, I think we end up in a situation where they're much worse off because we've sort of steered them to some place that we don't even really understand. And so what we are suggesting is that using an idea about, like, thinking about instead how those drugs shape these populations over time. If we understood that, if we thought about that a priori, instead of just saying, what's the most effective drug up front? Because right now our definition of effective is how much does it kill? How much does it kill of your tumor? How much does it kill of your infection? But if we thought instead, how does it shape the genotype? How does it shape the population? And how does that going to affect what's next down the line? I think we'd be better off, especially in cancer when we know the first drug's not going to cure them. In antibiotics, maybe it makes a little sense to do haphazard. Well, it's never good to do haphazard. But right now, most of the time you go to the doc, you get an antibiotic, you get better. But in cancer, you have disseminated disease, you get the first drug, you're going to fail. So I think we really need to be thinking. Go ahead.
I guess I'm wondering where it seems like there would be an inherent trade-off then between wanting to, between having, trying to maximize the chance of the next drug working.
Yes.
And trying to
yes.
get to the best possible landscape.
Yes, absolutely. There's a trade-off there that is certainly down, is like a next obvious step. No one yet has even accepted the idea that I'm selling, which is that we should think about how the first one's shaping the population. But you're totally right. And so if 5 years from now people actually listen to me, or I'm not proven wrong between now and then, which I probably will be, I think that's an incredibly important next step, which is you can't, so if someone comes in like that gentleman on the left, right, who's got 6 weeks to live, I can't afford not to give an effective drug. I can't afford to be like, hey, don't worry, this drug's not going to do much, but it's going to shape your tumor. It's like, well, I'm dying here. But. So there has to be some trade-off between effectiveness and the shaping. Yeah, it's a good one. Okay, so...
Well, would you give a person, I guess, a drug that wouldn't extend their life the longest? I mean, isn't that sort of like a conflict of interest?
Yeah, so I guess what I'm hoping through this line of reasoning is that if this does work, I would hope it's like a Queen's Gambit in chess, right? The first move in the Queen's Gambit, or the first 2 moves, you end up being down. You end up losing. You're down a pawn almost immediately. But you get tempo from it, and as long as you can get through the middle game, that tempo will see you through. I feel like that's sort of what I'm aiming for here, which is to say, you're right, if the upfront situation is dire, I can't afford to do this. But if I have some time to play with, I think in the long run this might be a better strategy. Okay, so the next thing I'm going to show you a kind of cool experiment that we did that I think brings some of these things home, and it's fun. So we had this idea we've already played with, where we sort of have the possibility of getting some population of bugs, giving them a drug or 2 or 3, and then testing how resistant or sensitive that drug is compared to just giving it the other drug, right? So some sequence first, best drug first, compare the two, get some resistance. And those were those plots we were looking at earlier, right? So now we had this sort of experimental idea. So we've seen in previous studies as effective sequences that they all had really small numbers of replicates. So believe it or not, that study I showed you in Science Translational Medicine was like 2 or 3 replicates. And there's actually pretty wide variability. And I've talked to buddies on the conference trail, and they've all said, I can't reproduce those results. I can't get them. I can't do that if I get them again. I can't do the same experiments and get the same results. And I don't think it's because the original group was trying to defraud us. But what I think is instead is that if you think about these landscapes, there's a lot of opportunities for branching points to happen. So I think it's a feature, not a bug, that these things occur. So what we did was, I'm going to skip that one. If we take those same 15 landscapes I showed you earlier that were really measured, and we do the same experiment, we just look at all the first drug evolution and then all the second drug evolution. But we do lots and lots and lots and lots and lots of stochastic iterations of that. We find 83,000 unique results. So it's not surprising that no one can reproduce their exact matrix. But we also find stark differences between the sort of best case and worst case. And when I say best case and worst case, I mean the most induced sensitivity and the least induced sensitivity. The most blue and the least, and the most red or the least blue. And this is the exact same drugs, exact same experiments, just those probabilities. And I think it lends us, and this is sort of our, if you do an expectation, this is where you end up. And so I think people have been working so far to be able to predict exactly if I give this one and then this one remember I had down here before that sensitivity bar, where I could say, oh, you're going to be 16 times more sensitive. I don't think that's what we should be thinking about. I think instead we should be thinking about what is the likelihood that I'm at least not worse. And so what we're moving toward is that concept we're calling a collateral sensitivity likelihood. I'm going to show it to you. We're going to come back to the experiment. We're thinking about instead codifying it as likelihood of being not worse. And so I'm not going to hurt my patient. Might I do a whole bunch better? Sure, it might. But I'm not trying to tell you you might do a whole lot better. I'm trying to tell you you might at least not do worse. And so with probability 1 in this very simplified model system, I can at least say that after cefprozil, ampicillin will be better than it would have been up front. I can't tell you how much better, but I can tell you better. And so this is a map of the likelihoods on 0 to 1, and then if you crank it, just look at the ones, and of course we can dial this to the level. So this is sort of where we want to go. And I'm going to show you a simple experiment we did, which somehow I put in front of this slide-wise, to test this. So I have a collaborator named Robert, who is actually my favorite professor during med school. It was kind of fun to be working with him 15 years later. It's really satisfying. And he also thinks that he thinks I'm crazy, but crazy enough that he's willing to do some experiments with me. So we did the following experiment. Sort of the same as we've been talking about. One strain of E. coli on a dish. These dishes are special because physics is so beautiful. These are called gradient plates. What you do is you take a regular petri dish, and when you grow bacteria, you have to dump in like a soup broth that becomes hard. So you heat it up, it becomes liquid, you dump it in, and then it hardens like Jell-O, and it's full of stuff the bacteria can eat. You can also mix drugs into that. You can do anything you want. It's like Jell-O. You mix vodka in. Although it would taste terrible because it'd be like beef bouillon or something. But I'm sure they do that in labs. So what we did with this experiment is we made a regular broth, and then we dripped in the center a whole bunch of drug. In this case, cefotaxime. And then we basically put it on a record player and spun it around and around and around, and we let physics drag the drug out. And so in the very center, we got a very high concentration, and on the edges, we have a very low concentration. So it ends up being like that video. We watched this really cool video that I can show you guys all later, but the day after I got these experiments, a science paper came out, of course, with like this super cool video of this in real time, and it was just like, So then we did, this is cool because it's cheap too, because you can seed some here, some here, some here, and some here from the same brush, right? These are the same bugs. Boom, 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 boom. And then you can let them grow overnight, and you have 4 experiments per dish. So look at this, just 3 dishes, so 12 replicates. And then we look at those 12 evolutionary replicates grown over the course of 10 days, and this is a fitness measure of each day. We only measure the fitness every other day. But you see, in just these 12 replicates, this huge heterogeneity in fitness, and you can see it right here. You can see, look at these fellas way out front, and these fellas way behind, and everything in between. You do this over and over again, and we actually find, if you then sequence them, you can actually find that these both had actually the same mutation. Oh, yeah. Totally separate bugs finding the same solution, which is a really good solution. And then these guys, or whatever, gender neutral, found a different one and did not quite as well, and yet a different one not quite as well. Now, what's the next question I'm going to ask? How do each of these mutants behave against some other drug compared to their, right? So that's the next question. And my hypothesis, of course, is that it's going to be all over the shop. And it is. And so here are 12 experimental replicates, all grown in the same drug, and then they're just tested against 4 other drugs in the same class. And we see striking examples of sensitivity, high sensitivity and high resistance in just the guy who, or gal who was evolving next door. And I think this result really is driving us toward this idea of moving away from the collateral sensitivity map and moving toward the idea of likelihoods. So that's sort of the striking idea. I'm going to flash a paper up because it was published yesterday, which I think is cool, where I'm now like, this is a frustrating part about science. Like if you're trying to do both theory and experiments, your theory gets ahead of your experiments because they take a really long time. And if you're hot, you know how that feeling when you're like killing it? And you can do like 6 years worth of work in 2 weeks, and then the next 6 years like nothing happens. We had this like explosion of excitement in the lab theoretically during the time when some experiments were happening. So basically what we did was so evolution's not repeatable, it doesn't commute, you can't make these maps. And we then, I'm just going to show you, I'm going to skip a few slides because it's been a fun discussion and I've been going slower and I want to show you some fun stuff with Gabe here in the last 10 minutes. So I'm going to skip about 6 slides, but I'm going to show you one of them because it sort of shows why. So we did this paper, which was published yesterday, where we did basically exactly the same experiment that Science Translational Medicine paper was, that I hope I've now convinced you was a terrible idea. We did it in cancer cells, and I don't even believe my own results anymore because of all the theory I showed you and the working bacteria. But I just basically cheated off them. I made sensitivity networks, I built the cycles, I sort of did the whole exercise, and it got me a paper, but don't read it. I mean, you can read it, but I don't believe it anymore either. So it's good that it informed my...
But if I'm getting it, you're saying that this other way of thinking that originally inspired you is too deterministic, that these things are more stochastic than you originally thought.
Yeah, and I think the important part is to qualify the stochasticity more than nail down these deterministic effects. I guess don't even think I can believe the deterministic versions. And what I really want to do is qualify the stochasticity within those processes. And I think for me that's where it's exciting at this point. But that experiment I did, it took like literally 5 months of doing all the, because cancer cells evolve much more slowly. So it took 5 months to do all the evolution, and then it was okay. So now I'm going to skip to a different model. I also, if anyone's interested, we also coded up like an individual stochastic-based version of that model that I showed you. And I'm going to show you 2 little movies because they're cool. So now, same idea, except I no longer require them to be the same population all the time. They can now spread out wherever they want on these landscapes. And now it's a birth-death process. You sort of start with a wild type. I mean, this is step 2, but they're sort of spreading out to their one-step neighbors. The size of the dot on this graph is going to be based on the population at that dot, and then blue means there's nobody there. Down here, you're going to see a trace of every single vertex through time. Here you're going to see the idea of allele frequency, so how many of them have a 1 in the first spot, how many have a 1 in the second spot, and so on. And up here is just an entropy measure. and this is through time with an evolving population. We can actually measure this right now in people through time during evolution under a selective pressure. We're getting close to being able to measure this.
So the two extremes on entropy are if it's a uniform distribution over the nodes versus all concentrated at one node.
Exactly.
Yeah.
And what I want to do mathematically, and I can't figure out how to do yet, I've been working on it for like a year and I'm just stuck. So help. Is I want to know if I can go from here to the landscape. So what I'm not showing here is that this is some landscape with hills and valleys. It is coded in already, right? I wrote the code, I know how it works. Some stochastic process here, and here's the dynamic trace. So the population proceeds through some, I'm going to show you this one because it's cooler. Population proceeds through some percolation, if you will, or evolutionary process, if you will, and you go through. And my question is if I know the initial state and I know these dynamics, can somebody in the room tell me what the landscape looks like? I can't do it yet. We're working on it. My friend Pete Jevons, who's a computer scientist at Oxford, is helping, and then I've got a postdoc called Nara, who looks usually happier than that, and a med student, Nikhil, who looks usually sadder than that, working on it, and we're really beating our heads against the wall. And I feel bad because I can't help any more than I've been trying. So one more cool little snapshot video, and then the rest of the talk is a little fluffy about game theory. So here's 2 videos in a row. I'm giving 2 drugs, ampicillin, sulbactam, which we've named SAM, and then just ampicillin. And what's going to happen is you're going to watch, and at time step 20,000, the landscape's going to change from this one to this one. You're not going to see anything happen at time step 20,000 because they share a peak. So it's kind of a boring one, but it's on purpose. Ta-da! If instead we stick in another drug in the middle, which is called cefprozil, same first drug, same last drug, new one in the middle. At time step 20,000, we get this exciting dynamics under the second drug as it reorganizes, and then under the third drug, we now end up at a totally different place. So this is, I think, the example, illustration of our hypothesis that you guys probably already all saw because math. But so that's the question. Can we go from these dynamics back to something? Because we can't really measure all the landscapes of all these drugs because, I mean, how do you do that, right? Remember that where I got this data, or these data, was like taking a lab strain and then like carefully permuting each base pair, and then, you know, even to do 5 took somebody like 4 years. So then how do you do that for every patient on their whole genome? Like, you just can't. So the questions here are myriad, which is, you know, how much information do I need? How many alleles is too few to consider? Because clearly if there was some like completely dominant allele that I'm not considering, who cares what happens here, right? Just go to the peak that we're not even modeling. But then too many and it becomes intractable. So I think I'm stuck. So I've moved on to other problems because I can't figure it out. Okay, so now this is the part I'm going to skip because I don't believe it anymore. So the last 6 or so slides are playing around with this model situation called evolutionary game theory. And so I think many are probably familiar with it. The idea is it's an ODE system that's constrained by some assumptions. And I'm not going to go into the weeds of the modeling formalism because you don't need me to, and I don't have time, and that's not the point either. The point is that I work with some fun people. David's a game theorist, Andre's a cell biologist, Jeff's a medical student, and Artem is a computer scientist. He's a graduate student of mine, and he's like the most difficult person to work with ever, but he's also the smartest. And he's listening, or he will listen to this recording. Hi, Artem! And we've had a lot of fun. So in this experimental system, I'm going to describe, what we do is we took advantage of some experiments we already did. For that paper that I whizzed by, in that paper, we evolved resistance, just like in the just like in the bacteria stuff, we evolved resistance to a bunch of different drugs. So we had like a wild type, and then we had a bunch of different resistant ones we could play with. So what we did was we already had all these strains laying around. So Andre, through the magic of cell biology, turned the parental ones green, and he took the resistant ones and made them red. And now all of a sudden, under the microscope, there's competition. And we also know from other literature that these special cells called fibroblasts, which are not cancer, they're like part of us that respond to inflammation, really affect the dynamics between these cells, especially when drugs are involved. So we know that cancer-associated fibroblasts can rescue sensitive cells from being killed. So game theory is a lot of fun. I've been playing with it for some years. David and Sandy and I wrote a paper in 2011 where we found out this cool idea that timing and order of therapy matter, so we can write down some dynamics. Don't pay attention too much. The idea is fun. If you know the payoff matrix, you can do lots of fun stuff mathematically. And you can say, oh, well, giving a drug for a little bit of time is not as good as giving for a lot of time, because here our green ones win and here our blue ones win. So we can perturb the dynamics, we can qualitatively shift how the tumor evolves. But this is all predicated on knowing payoff matrix. And well, we made that up. And people accepted it because they're like, oh, he's a doctor. Well, come on, I just made it up. We wrote a paper, Artem wrote this paper with more exciting math just this year, 2017, version of cancer, same idea. We took some evolutionary game dynamics, which frankly are arbitrary. We invented them. We think they make sense. We feel good about the story we told with them. And in this one, we say that the order of therapy matters, which is what I just told you in the last 45 minutes. But different model system. But again, it's predicated on knowing what the equations look like. And what do the equations look like? Well, we made them up. So I would argue that that's not good enough. And I think that what we're going to try to do is use an experimental system that we carefully design to back out that payoff matrix. If we can do that, then we can use all these fun tools we've been developing for a few years to do something more interesting and maybe make predictions about an in vitro system, or maybe make predictions about a patient system. So 5 minutes left. We took the sensitive cells and the resistant cells and we did a sanity check. This is an experiment where these are the parental. The red are the H3122, which is just the name of the parental. Blue are the ones that we evolve resistance to over 4 months. And so they're ALK resistant, so this ALK is a kind of a drug. This is no drug, so our parental is way more fit. This is fitness over here. It's a proxy, it's called optical density. It just says how much they grow in a tube. So much more fit with no drug, makes sense. There's a cost to resistance. And then lots of drug and it flips. So we have this cool sort of cross, the opportunity for some, over here there's one competition dynamic and over here there's another competition dynamic. We sort of write down a simplified version of the replicator equation such that we actually can look at something we're going to call a gain function, which is basically a single functional form that tells you how much one is competing better than the other. So it's like a normalized version of fitness. And then we're going to play out some evolutionary dynamics. So first thing we did was this is just the sensitive cells. They're green, and this is lots of the drug that they're sensitive to. Here's the same sensitive cells with, don't be confused, these are actually fibroblasts in this one, not the resistant cells. What we find is that if you grow these out over a few days or weeks, there's sort of more green over here at the end of the experiment than over there. So these fibroblasts provide some sort of boost where they allow the cells to escape the drug. Same amount of drug on both sides. Okay, that's nifty and fun. What we then did is we wrote down a computer vision algorithm. We took a picture of these plates every hour over the course of that many days, counted the number of reds and greens with some clever short thresholding, and we're able to get dynamical traces out of them. We can then fit exponential functions, and we can do this experiment many, 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 many times. So hold on, this is just another movie of the same thing, red and green together to show that we're sort of modeling the same thing. So now I'm going to show you this plot first. So now what we can do to read this plot, what you do is you say, so we did something like, so every one of these dots is 3 experiments, and each one of them is a different initial condition. So if you're a game theorist, what matters is frequency dependence. So if you have 10% of one and 90% of another, the dynamics will be slightly different than if you have 50-50. So we actually set up an experiment for almost every possible combination of initial conditions. This is what having medical students is great for, because this is horrible. And what we're able to do is then understand as you change the initial proportion of parental, so over here it's all parental, and over here it's no parental. As you change that initial proportion, you can see the growth rate is actually changing. So this is the growth, this is the exponential curve, the constant. You can see that this curve changes, and And we have 4 conditions. This is no drugs and no fibroblasts, so just the cells in competition. This is no drug but cells and fibroblasts, all the cells and all the fibroblasts. And this is drug and fibroblasts, and drug and no drug. So all the possible ons and offs. And what we find is that the game that's being played actually shifts in this one condition. So over in all 3 of these conditions, we have a sort of canonical game called Deadlock, which is actually kind of a boring game. It only has a single steady state. As I said, it's an isomorphic steady state where all the resistant wins. However, here, in the presence of no drug but with the fibroblasts, we have an evolutionary steady state where there's a coexistence possible. So we see sort of this idea that by changing these environmental conditions, drugs and fibroblast presence, we're actually able to change the way the game looks and actually able to change the payoff matrix itself. And so this slide is busy. This is our figure 1 for a paper, and it just shows really the same thing. It shows we take the microscope slide over the course of time, fit it to this is a semi-log plot to its exponential growth, and we're able to understand how these dynamics play out. And then we're able to, from that, back out the exact, these are just scaled by, you can multiply by a constant and not lose anything. So if you read back to those plots 2 slides ago, simply the slope and intercept gives you these numbers exactly. So we can actually figure out the exact game that's being played with each scenario. So this is a kind of boring game that we found that was being played, but I think the point is that we're able to measure it, able to put it back into this format. It looks like there is a competition that game theory would predict, and we're hoping to try to extend this to a 3-player scenario, and then really try to make some predictions going forward about how our perturbations might change stuff. So that's fun. I think I showed you we use a bunch of tools. This is a CAT scan of a young man who I'm taking care of right now. He actually, I have been taken care of, I should say. Matt died last week. I want to show you what his lungs look like. So this is a CAT scan of his lungs. All those things that you saw that weren't black, so this is his liver, these are his kidneys, kidney, liver, guts, spleen. Lung is black, all those white spots are tumors. That's his heart, he's supposed to have that. So he was full, and everything we gave him didn't work. And it was really a bummer, but that's why we're doing this. It's because you're not supposed to have lungs that look like that. So anyways, that's my talk. Group, thanks to PhD supervisors who have been super patient with me. And then my team, do you guys remember Voltron? Yeah? They just recently redid Voltron. Turns out Voltron's this awesome robot that defends the universe. And he's made up of 5 really cool lions that are also robots that they have like pilots. And like when like a not so important threat to the universe shows up, the lions just like go out there and take care of it. Like maybe the red lion will be like, hey, you get this one. The red lion will get in and go out. But when something really terrible shows up, all the lions have to come together and form this badass robot with a sword that comes out of nowhere. And then the robot is even tougher. And so in my lab, Nara's the green lion, Drew is the yellow lion, Andrew's the blue lion, Artem, of course, being difficult not having his lion, but he's red. And then, of course, I get the cool one, which forms the head, as the black lion. And then that's how we go about science. It's a team effort is the point. So that's my talk. Questions that weren't asked, sir?
Can I ask you something a bit tangential to your talk?
Please.
About drug scheduling.
Yeah.
So with antibiotics, I always find it strange that when you go to the doctors, they tell you how important it is to take an antibiotic every day. But then when my father had chemo, he had his chemo every 3 to 5 weeks. And they were very slack about when he scheduled his chemo. Does drug resistance get affected a lot by when the drug is taken and the schedules that are formed? And should the UK be less slack about when they schedule chemos?
Yeah. So the paper I skipped is all about drug holidays, which is how long you're not on it. And it's, so this figure here is exactly your question. So if you wait 1 day and measure the collateral sensitivities, it's different than waiting 3 days, it's different than waiting 7 days. They're all different, and we have no idea. And so yeah, it's incredibly important. The problem is right now we have no idea what we're doing. Let me walk that back. We're doing as best we can, but it's a really hard problem, and I don't think people are thinking about it this way yet. Except me, just shouting in the wind. But I think that it's incredibly important, and I think these evolutionary changes. So basically, you give a drug, right? It's, in my thought process, you have the population evolving on a landscape. You pull off the drug, and it's not like everything stops. There's some new landscape, which is the patient with no drug. And that's like maybe the original landscape. Like maybe the original landscape the tumor is growing in to start. But might also be different because you've now given that patient chemotherapy. Their immune system's changed. Their normal tissue's changed. We don't know that. We don't know anything about that. And I think that, to the best of our knowledge now, those times don't matter very much. But this paper, I think, opens the door to asking the question about time off. Because actually we find that there's certain windows in that time off where some drugs pop on and off for sensitivity. So at 14 days, you're incredibly sensitive to this drug, whereas at 7 days, it was this one. And we just don't know. And I think, like I said, this experiment was not done as well as I would do it now, but I think it opens the door. So yeah, but I don't think we have better information right now. Please.
If you think of cancer as being a population that contains resistant and non-resistant cells to a particular drug, and the traditional treatment strategies are killing off the non-resistant cells, and therefore allowing the resistant cells to proliferate more. So in your studies, does it say anything about sort of to what extent you should look at the population dynamics of these things so that you allow, in normal conditions, if the non-resistant cells tend to outcompete the resistant ones, then you want to take advantage of that.
Yeah.
And you want to be very cautious about how much drug you apply. You want to control things, but you don't necessarily want to try to kill everything. You're not going to succeed, and when you do that, you're going to create the conditions that's going to allow for the resistant cells to dominate.
Yeah, so I think what you're talking about is this idea of competitive release. And it's sort of, the idea is sort of here, where like you just said, in the presence of drug, one might outcompete the other, and when you pull the drug off, the table's flipped. And so the question then is, can you think about a strategy where you sequence the drugs in such a way that you maximize the competition that they experience on their own, so they maybe slow each other down, and that you also kill them? And absolutely we thought about that, and Nara, the unhappy looking but actually happy postdoc I have working with me, is working directly on that in a really simple 2 ODE system, which I'd be happy to share with you. And I think that some of my previous supervisors are trying that in a clinical trial now, using a really simple summary metric of tumor growth. And I think that there's a lot that we can do with that simple idea. So we thought a lot about it in this sort of landscape idea where we have more heterogeneity than just the 2 types. We could think about it as, you know, a low peak and a high peak competing against each other as they kind of switch when the landscape changes. There's a lot of ways to think about that. But yeah, certainly I think that's a key point that we're working on in a simple system that I didn't talk about today.
Just to follow up that point, is there a way to think about this instead of killing the cancer? Actually, like, we want to help the resistant bacteria, like, actually take over, and then at some point we just kill them off.
Yeah, so I think that's sort of what I'm trying to suggest with the shaping idea, right? I'm sort of trying to say that if I knew there was a certain phenotype that I had no trouble killing, if I could promote it to starting, and then you put that together with the idea that John brought up, you know, what if that also outcompeted that sort of resistant one to start? You know, I think all these concepts are central to this, but it's a matter of putting them together in a sensible way.
And do you study quiescence by any chance? Because that actually seems like it would not help in some sense.
Yeah, absolutely. Yeah, lots of things go dormant. So again, in the paper that I didn't talk about, I we took like 4 months to get resistance. If you look in the middle of that time period, there's actually cells that don't grow. They're just sitting there. And they're not good at resisting the drug in that you give them drug. They're good at resisting the drug in that they don't die, but they're not good, like they can't grow in the presence of drug. But they're good enough to sit there and chill, and then you wait, and you wait, and you wait, and you wait. Some of them figure out how to grow. So it might be that there's an intermediate sort of persist or quiescent state that actually precedes. And I don't know if that's like, I'm imagining, I think of everything like these landscapes, so I don't know if that's some sort of neutral space. When I say neutral space, I mean connected component or so connected subgraph where it's all equal fitness, but it's sort of walking randomly around that neutral area. It's like fit enough not to die, but not fit enough to grow. But maybe it's walking around randomly and then has some sort of right-skew distribution in which it finds the next mutation. I don't know. But yeah, if we think about it, it's just hard to put all the things in, right? Sir?
So I think I have a question which rounds up several of the questions you already answered. You're clearly talking about an optimization problem. But part of the reason you're getting those questions you just got was that you never heard what the objective function is. Are you trying to maximizing the probability that the patient will live a certain amount of time? Are you trying to maximize the life expectancy, minimize the number of the expected number of cancer cells? What's really going on?
Yeah, I don't know. I think that's similar to, it's central to this whole thing, right? Sort of like fitness is central, but I can't tell you what it is. And I don't know what the objective function should be. I mean, it should be survival, in my opinion. But how do I map that stuff to patient survival? You know, simply, or like the naughty model, as my UK colleagues would say, would be to say, okay, well, some threshold number, 10 to the 10, and now the patient dies. But that's not always the case. It might be 10 to the 7 for some people. It might be 10 to the 12 for other people. You know, I've personally taken tumors out that are like 4 liters. That's going to be, you know, 10 to the, I don't know what that number is. I don't even know if a number is the right thing. And so I don't know. And I think maybe, I don't know. Anyone else? Can anyone help me here? I'm waffling. I'm waving my hands like crazy.
Well, one thing before everyone goes off to eat at refreshments, you mentioned that movie that came out right before your study. I think people here would like to see it if they haven't seen it before.
It's like 1 minute. It's pretty cool. Does this thing have sound?
You could plug in something with sound, but you don't need it.
You don't need it.
Oh,
That's okay. We'll take
you got it with your edgy
edgy room. We'll pick it up. And it's right here once the dinosaur goes away. Have anyone ever figured out why there's a dinosaur on this page?
If you click on it, you can play a game.
No way! That made the whole trip worthwhile. Thinking about it.
Oh.
It'll get there. It's just pretty good.
So the infinite building is basically a petri dish, except that it's 2 feet by 4 feet. And the way we set it up is that there are 9 bands, and at the base of each of these bands, you put a normal petri dish thick agar with different amounts of antibiotic. On the outside, there's no antibiotic, just in from that. There's barely more than the E. coli can survive. Inside of that, there's 10 times as much, 100 times, and then finally the middle band is 1,000 times as much antibiotic. And then across the top of it, of course, thin agar that bacteria can move around in. The background is black because they're ink in it, and the bacteria appear as white. First, you see they spread in the area where there's no antibiotic, up until the point they can no longer survive. Then a mutant appears on the right. It's resistant to the antibiotic and spreads until it starts to compete with other mutants around it. When these mutants hit the next boundary, they too have to pause and develop new mutations to make it into 10 times as much antibiotic. Then you see the different mutants repeat this at 100. And after about 11 days, they finally make it into 1,000 times as much antibiotic as the wild type can survive. And so we can see by this process of accumulating successive mutations that bacteria, which are normally sensitive to an antibiotic, can evolve resistance to extremely high concentrations in a short period of time.
See how bummed out I was. I came out the day after my little circle plate.
All right, well,
What did you say if we told you?
so let's thank Jake again.